This video provides an introduction to immunology, the study of host defense mechanisms. Immunology is the study of host defense mechanisms. Immunity is the ability of a host to protect itself against pathogenic foreign organisms. The immune system includes the tissues, cells, and molecules that mount the immune response. There are two types of immune systems: natural or innate, and adaptive or acquired. The innate system, present in all living creatures, includes barriers like skin, tears, and saliva. The adaptive system is specific, has memory, and involves specialized cells, cytokines, and antibodies. Defense mechanisms are categorized as either innate immunity or acquired immunity. Immunogenicity is the capacity to induce an immune response, typically by foreign, complex, high molecular weight compounds like proteins. Antigenicity is the ability to bind to antibodies or immune cells, which may or may not lead to an immune response. All immunogens are antigens, but not all antigens are immunogens. The innate immune system is a reliable first line of defense against many extracellular organisms and is a property of every living organism. However, it cannot deal with all intracellular organisms. It is nonspecific and has no memory. The innate immune system is also known as the natural or native immune system. It is rapidly mobilized, nonspecific, and not dependent on prior exposure to a foreign invader. However, it may not be sufficient to prevent foreign material from persisting in the host. Components of innate immunity include physicochemical barriers, molecules in body fluids such as lysozyme and complement, and phagocytic and cytotoxic cells like neutrophils, macrophages, and natural killer cells. This slide is blank and contains no additional information. Threat neutralization by the innate immune system often involves the respiratory or oxidative burst. This process, mediated by membrane-bound NADPH oxidase and other enzymes, produces substances like superoxide anion, hypochlorous acid, hydrogen peroxide, and chloramines. The acquired immune system is specific and has immunological memory. It involves dedicated immune cells, such as lymphocytes, and molecules like antibodies or immunoglobulins that specifically counteract antigens. It also includes specific barrier immune systems like mucosa-associated lymphoid tissue and gut-associated lymphoid tissue. Antigen specificity, mediated by lymphocytes, is the single most important aspect of the acquired immune system. Each clone of a lymphoid cell responds to only one antigen. T cells handle surface-bound processed antigen, while B cells deal with soluble, extracellular native antigen. Specific immunity is humoral when antibodies remove the antigen, and cell-mediated when T cells and macrophages are involved in pathogen clearance. Stem cells in the bone marrow differentiate into B cells for humoral immunity and T cells, maturing in the thymus, for cell-mediated immunity. Immunity after infection is termed active immunity. Immunity can be transferred passively by antibodies or cells, such as through breast milk or maternal IgG antibodies via the placenta. Vaccination can be passive, using immunoglobulins, or active, using antigen or attenuated organisms. The primary immune response begins the development of acquired immunity. This involves an afferent phase with antigen-presenting cells trapping antigens, leading to T cell transformation. The effector phase involves activated T cells, inducing other cells like B cells and macrophages by secreting cytokines. The primary response includes the appearance of antigen-specific T cells and Ig-secreting B cells. In the secondary immune response, upon subsequent exposure to the same antigen, memory T and B cells are recruited faster and more efficiently, leading to much higher immunoglobulin levels. This slide illustrates the antibody response following exposure to an antigen. T cells develop in the bone marrow and mature in the thymus before migrating to lymphoid organs. They are key cellular components of immunity, possessing an antigen receptor that recognizes specific intracellular antigens via the T cell receptor. This receptor only recognizes antigens combined with major histocompatibility complex, or MHC, proteins on the cell surface. T cells only recognize antigens when they are associated with MHC molecules on the surface of cells. Clonal selection increases the number of effector T cells to destroy invaders. T cells regulate the proliferation and activity of other immune cells, like B cells and macrophages. They provide defense against pathogens, cancer cells, and transplanted tissue. Unlike humoral immunity, cell-mediated immunity is not passively transferred. This figure illustrates the T lymphocyte and natural killer cell-specific responses. The types of T cells include helper T cells, which have a central role in the immune response, cytotoxic T cells, which destroy target cells on contact, delayed hypersensitivity T cells, involved in allergic reactions, and suppressor T cells, now called regulatory T cells, which shut down the immune response. CD4+ cells recognize antigens presented by antigen-presenting cells. CD8+ cells recognize and kill virus-infected cells or altered self cells, such as tumor cells. CD4+ T helper cells interact with other cells by releasing cytokines to control the immune response. Th1 cells activate macrophages to destroy pathogens, while Th2 cells help B cells produce antibodies. B lymphocytes are specialized for producing immunoglobulins after differentiation into plasma cells. They control pyogenic bacteria, prevent blood-borne infections, and neutralize toxins. B lymphocytes constitute about 12% of total lymphocytes. B lymphocytes can respond to peptide, carbohydrate, and glycolipids. They usually require T cell help to respond to antigens, but can also recognize antigens directly through surface immunoglobulins. B cells mature and proliferate in lymph nodes. This slide is titled Ag-Specific Responses, but contains no body text. The 5 classes of immunoglobulins, or Ig, are IgM, IgA, IgD, IgG, and IgE. Immunoglobulins have a 4-chain structure, 2 light chains and 2 heavy chains, linked by disulfide bonds. The chains are coiled into domains, approximately 110 amino acids long, stabilized by an intrachain disulfide bond. This slide is blank and contains no additional information. The terminal regions of the heavy and light chains form the variable regions, where Ig combines with antigen. This variability accounts for the wide range of antigen specificity, known as antibody diversity. The other domains are the constant regions. Fragments like Fab and Fc have been identified using enzymes. The Fc regions, formed from heavy chains, determine the isotype and biological functions, such as opsonization, complement activation, and ADCC, distinct from antigen binding. The hinge region also affects function. IgG, immunoglobulin gamma, neutralizes toxins, activates complement, and is involved in opsonization. It is the only Ig that can cross the placental barrier. It has 4 subclasses and is present in the highest concentration in the blood. IgA, immunoglobulin alpha, exists in monomeric and dimeric forms. The dimeric form is secretory IgA, found in secretions like tears, saliva, and breast milk. It is an important antiviral Ig and the signature molecule of mucosal immunity. IgM, immunoglobulin mu, is pentameric, a good agglutinator, and effective at activating complement. It is the first class of antibodies synthesized by B cells after antigen exposure. It is also known as MIG and has 10 antigen binding sites. This slide is blank and contains no additional information. IgE, immunoglobulin epsilon, is active against parasitic infections, especially helminths. Its Fc portion binds to receptors on mast cells and basophils, triggering mast cell degranulation. It is found in high concentrations in atopic individuals and mediates type 1 hypersensitivity reactions, like asthma. Major histocompatibility complex, or MHC, class 1 molecules are expressed on all nucleated cells. Class 2 molecules are expressed on antigen-presenting cells, and their expression can be induced on other cells during immune reactions by interferon gamma. Cytokines are short-range, multifunctional, and short-acting mediators of cellular activities, particularly within the immune system. They are primarily released by activated T cells and other immune or non-immune cells, acting in an autocrine or paracrine manner. The 4 major classes of cytokines are interleukins, interferons, colony-stimulating factors, and tumor necrosis factors. Natural killer, or NK, cells are part of the lymphoid system but lack differentiating surface markers. They are large granular lymphocytes, particularly effective against virus-infected and tumor cells. They kill via granzymes and perforins. Antigen-presenting cells, or APCs, capture the antigen at the site of inflammation and transport it to regional lymph nodes or the spleen. Here, the antigen is presented to T and B cells to initiate the acquired immune response. Examples of APCs include macrophages, B cells, and dendritic cells. Macrophages and B cells recognize antigen through the Ig molecule. Dendritic cells process and present antigen to naive T cells via MHC class II and are considered the most potent APCs. This diagram illustrates antigen recognition by a CD4+ T cell presented by an APC such as a macrophage, via MHC class II. The complement system includes at least 9 plasma proteins and regulatory factors that mediate inflammatory processes. These components are synthesized by macrophages or hepatocytes, circulate as inactive proenzymes, and are heat labile.